So HAT, which is a histone transacetylase. And HDAC. And HDAC stands for histone deacetylase. Deacetylase. So those are the enzymes. So those enzymes regulate whether or not that DNA is available for the binding of the proteins at the promoter region. So this is another level of regulation, right? Is everybody still with me? Now these proteins, these HAT and HDASs, are contained within structures that we call chromatin remodeling complexes, CRCs, chromatin remodeling complexes, because this complex is able to remodel the structure of chromatin. We can't talk about everything they do, but we talk about one thing they do, and that is that these remodeling complexes have co-activators and co-repressors associated with them. And the co-repressors, right, are the deacetylases that we talked about. HDAC is a co-repressor. You can see it deacetylating these tails, and that would cause them to come together. An activator would have deacetylase, the histone acetyltransferase, histone acetyltransferase, or histone transacetylase. Anyway, this guy acetylates those tails. When they're acetylated, they become less tightly associated with DNA. So they allow these things to breathe. Now, there are other things that these remodeling complexes can do. And I just want to tell you briefly what they can do. One of the things that they could do is they can perform a movement or a procedure that we call phasing of the nucleosomes. And what phasing is that their nucleosomes are able to move one way or the other slightly on the DNA. There's nothing specific about the DNA that's wrapped around those nucleosomes. Nothing very specific about them, about that wrapping. There's some physical stuff, but not sequence stuff. Okay, so DNA randomly wraps. If you take a nucleosome and throw it into a viral cell that doesn't have nucleosomes, the DNA will wrap. It just happens. So anyway, there's nothing very specific about these that does that. But they're able to move these a little bit. The other thing is that these things have nucleosome exchange molecules. And what they can do is they can take a nucleosome, right? Remember this core with the 8 proteins? And they can exchange or some of the histones in there. So they'll exchange some of the histones for a very specific variety of histone, a very specific histone that doesn't wrap as tightly around the DNA, right? It allows it to come away. They can also make 2 other maneuvers that we think they can make. One of them is we think that they can take and they can remove half of the histone or half of the nucleosome. So they can take out 4 and leave 4 histones in there. Sort of leave a half moon, a half nucleosome in there. And then, of course, the DNA doesn't interact. And then finally, there are some that actually can take these nucleosomes, remodeling complexes, can take them and move them right on out, right? So that DNA becomes very loose, very accessible, very either transcribable or replicable, right? Same problem with DNA replication. You have to loosen up that DNA so the enzymes can get in there and do their thing. And the remodeling complexes can put these things back. So kind of the idea is that remember when we transcribe, we're going to have a polymerase molecule that moves on down here, right? It's going to move on down here and transcribe through here. It has to loosen up these things, so in front of these transcription factors and polymerases, it's going to acetylate these guys and it's going to move on down and change the binding of this DNA around the nucleosome. And as the complex moves on the backside, it's going to rewrap it and leave it the way it found it. Okay? So transcription, we have to loosen up that DNA. We even have to around the nucleosome. We even have to modify the arrangement of nucleosomes with the DNA sometimes for access. And then, of course, once we do that, we have to have promoters recognize, or proteins recognize the promoters. Now, we talk about these 3 things sort of separately, but they sort of occur almost simultaneously. You can see here transcription factors associated with these repressor molecules. They're all kind of in this complex doing everything at once. Sometimes the repressors actually have to, or the CRCs, chromatin remodeling complexes, bind first and begin to modify DNA. They contain acetylases. They open up the DNA, so now the transcription factors and proteins can bind. And it's different. We can't say it's this way in one gene and not in another. Okay? Okay. So that's what we know now about loosening up and regulating and binding of transcription factors to eukaryotic promoters. Does anybody have a question? Because that's all we're going to talk about that. Okay. Now what I want to talk about is something that's ought to be a lot easier for you guys, because you probably study this system. Certainly some of you have looked at it in biology and so forth. You want to look at the regulation of gene expression, and we're going to look at the regulation in prokaryotic cells a little more carefully. We're going to look at the promoter, how regulation works, and how some of these binding proteins, what do the binding proteins do? You said that some of these binding proteins bind at the promoter, and if they're somehow regulating expression, we're going to look at how they do that. And we know the best examples we have that in prokaryotic cells, of course. So we're going to look at prokaryotic cells. We're going to look at the last outline, which you've all heard the story about before. Hopefully we'll talk a little more. So remember, in bacterial cells, they have their genes arranged in what we call polycistronic groupings. So you can see that here there's 1, 2, 3 structural genes, and they're all right next to each other. They are missing, they're spaced only by a stop codon and a start codon. When they're transcribed into RNA, they're transcribed as one single messenger RNA. The advantage of that is they're all regulated by one promoter. And generally, all of these genes that are in a polycistronic piece of DNA code for proteins that are all involved in one pathway. So all leucine biosynthesis, all galactose utilization, whatever. And that regulates these things with one promoter, and it regulates the stoichiometric amount of each of those that are made, so that you don't make a ton of this and a little bit of this if they all have to work in the same pathway. Okay? So it regulates them that way. So in E. coli, the expression or the use of galactose is very closely regulated. E. coli will grow, and if you put it in glucose and galactose, it'll grow on glucose. It loves to grow on glucose, right? Glucose is already just a single ring, and it just metabolizes the heck out of it, just like you guys learned. But often it's not available. You know, these cells don't have glucose. They use other sugars like galactose and some other things. So if you give cells galactose and glucose, they'll grow on the glucose and they'll ignore the galactose. If you put them only in galactose, then they'll grow on it. And the way they do is they turn on or they activate these proteins or these genes that are needed for the utilization of galactose. The genes are the Z gene, which codes for beta-galactosidase. That's the guy that actually takes the galactose and cleaves it into sugars that can be used by the cell. The LAC-Y gene is a permease. It's a protein, a membrane-bound protein. It's in the inner membrane, right, on the inside of the intermembranal space of E. coli. And it allows, it's a molecule that it's like a, what do you call those guys? I lost my name. It's a beta barrel, right? And it allows the cell to take up the lactose. So it's a permease. And then this LAC-A gene, we don't understand very well. The LAC-A is codes for transacetylase, which modifies galactose if it's not being used. Not terribly important in the pathway and the usage, at least not that we understand. So there are 3 proteins. They're all regulated by a single promoter, right? This region here. And that promoter has an operator. That's what the O stands for. We're going to talk about how that promoter and operator works. The operator actually determines whether or not this gene is expressed or not. And the gene is either repressed or activated. And when it's repressed, it's shut off. And it's repressed by a protein that's coded by the LAC-I gene. So here's the LAC-I gene. It has its own promoter. And when LAC-I is expressed, it transcribes a messenger RNA that codes for a protein that's about 40,000 daltons or so. And that protein forms a tetramer, and that tetramer binds to the operator region. So that this tetramer binds to the operator, and it represses expression, it stops expression. The way that the expression is activated is to remove the repressor from the operator. Make sense? So here's another picture that shows what's happening again. So here's the same opron. When the LAC-I gene is expressed, it makes a protein. That protein forms a tetramer and binds to the operator. What it prevents, you can see that the operator is kind of to the side, it's to the, what we call upstream, to the 3' side of the promoter, right? And remember, what happens at the promoter? What's going to happen at the promoter? Protease is going to bind, it's going to align, and it's going to start transcribing these genes, right? So now you put this protein in there at the operator on this side of the promoter, you just literally sterically block this thing. So the polymerase can't bind there and transcribe that gene. So it represses expression. How is it activated? Remember, these cells want to activate this set of genes when there's plenty of galactose around. So actually what happens is that when galactose is present, right, the inducer, the galactose binds to the repressor protein. So the repressor protein has a binding site. When that binding site is empty, it binds to the operator. When it binds to the lactose, it comes off of the operator, or it can't bind to the operator. If it's not bound, it can't bind, and if it is bound, it'll come off. So it comes off, that opens this up, and now the protein, or the polymerase can sit on there and transcribe these genes. Question? This is negative regulation, right? Because the repressor negatively regulates expression. When the repressor is on there, it negatively regulates the expression. Now this particular operon is also controlled positively.